Hi, everyone. My name is Troy.
And I'm Michelle, and we're from Group 8 covering the article titled Deregulation of Mitochondrial F1F0 ATP Synthase via OSCP in Alzheimer's Disease. Alzheimer's disease is the sixth leading cause of death of Americans and the fifth leading cause of death among Americans aged 65 and over. Therefore, it is a heavily studied disease in the field of neuroscience. Alzheimer's disease is characterized by memory loss and other cognitive impairments, which result in dementia and can eventually lead to death. In order to reduce the number of deaths by this disease, researchers are working hard to study the symptoms and pathological features in order to target them and prevent the disease from deteriorating and resulting in death. Previous research and clinical observations have highlighted mitochondrial dysfunction as one of the earliest pathological features of Alzheimer's. Impaired mitochondrial oxidative phosphorylation, also known as OXPHOS, shown above, has also been identified as a key feature in Alzheimer's disease individuals and animal models. In this article, researchers investigate impaired OXPHOS efficiency and its link to defects in the F1F0 ATP synthase. We understand that deregulation of the F1F0 ATP synthase shown via the GIF on the left is responsible for decreasing the quality oxidative phosphorylation and also increases the permeability of the mitochondria. These side effects are present in patients with Alzheimer's disease, but how exactly the mechanism occurs was unknown before this paper. This paper hypothesizes that oligomycin sensitivity conferring protein, hereafter referred to as OSCP, has physical interactions with amyloid beta, referred to as AB or alpha beta, whose cryo-EM is shown on the right, was causing F1F0 ATP synthase deregulation, leading to Alzheimer's. This paper further states that regulation of the mitochondrial F1F0 ATP synthase, OSCP, could help combat the effects of Alzheimer's disease via stabilization of the OSCP. This study utilized immunoblotting or Western blotting in order to compare the expression levels of the major subunits of mitochondrial F1F0 ATP synthase and protein extract from the temporal lobe of subjects. Proteins were separated by STS-Page and then transferred to a polyvinylidene difluoride membrane for blotting. The membrane was first incubated with primary antibodies and then secondary antibodies. Finally, images were collected on a Bio-Rad ChemiDoc imaging system. Immunoblotting is a method by which to identify or quantify the amount of specific protein within a given sample. First, gel electrophoresis takes place in order to separate biomolecules. The process utilizes a uniform electric field in which charged molecules migrate depending on their net charge and frictional coefficient. The next step is the immunoassay. This utilizes primary and secondary antibodies to identify and quantify biomolecules. A primary antibody binds directly to the antigen of interest, and a secondary antibody binds to the primary antibody. Finally, the results are viewed using fluorescent chemiluminescent detection imaging system and analyzed. As discovered in the article we previously covered, OSCP acts as the binding target for cyclophilin D, also known as CypD, and is instrumental in the F1F0 ATP synthase disconnecting from the C ring and the formation of the mitochondrial permeability formation pore. The release of the F1F0 complex can be prevented through the inhibition of CypD by introducing CSA as a CypD specific inhibitor. The abnormal buildup of amyloid beta and the subsequent amyloid beta toxicity plays an integral role in the pathophysiology of Alzheimer's disease-related mitochondrial synapse degradation. There is strong evidence to suggest that interaction between OSCP and amyloid beta impairs mitochondrial function through wider F1F0 ATP synthase dysfunction and instability. Abnormal MPTP formation resulting from amyloid beta OSCP interaction is responsible for a host of mitochondrial defects, including suppressed ATP synthesis, lowered mitochondrial membrane potential, and overall oxidative phosphorylation deficiency.
So the image on the left shows how by using immunoblotting to compare expression levels of F1F0 ATPase subunits in temporal lobes, they found that OSCP expression was slightly reduced in MCI brains, but greatly reduced in AD patients. However, expression of other subunits in the ATPase were not changed significantly in temporal lobes of subjects who had mild cognitive impairment or Alzheimer's. This finding implies that OSCP loss is most likely not a result of F1F0 ATPase reduction in Alzheimer's disease. As shown in Figure 2, synaptic mitochondria in amyloid beta-rich environments are more vulnerable to OSCP loss when compared to non-synaptic mitochondria. Alongside this, the other subunits in F1F0 ATPase had expression levels which remained unchanged, indicating that OSCP expression is selectively expressed alongside the manifestations of Alzheimer's disease symptoms in the mice. So the impact of OSCP loss was observed against the F1F0 ATPase's role in maintaining the mitochondrial membrane potential in ATP production. They found that decreased intensities of TMRM, an indicator of membrane potential, implied collapsed mitochondrial membrane potential, and they discovered a reduction in ATP generating, suggesting a regulation of mitochondrial oxphos efficiency. Shown in figure 1 on the left, the scientists found that although CSA suppressed MPTP formation in control neurons due to its inhibition of CYP-D, when MPTP activation was linked with OSCP loss, this activation was not blocked when CSA was added. However, they also found that OSCP loss resulting in MPTP activation was not protected through a decrease in CYP-D amounts. Based on this, the researchers were ultimately able to suggest that the activation of MPTP, which is induced by OSCP loss, does not depend on the status of functionality of CYP-D. Another observation they made while trying to observe the OSCP stabilization of the F1F0 complex is that they discovered an increase in MPTP formation is closely associated with F1F0 complex disruption. This informs us of how OSCP losing function triggers the MPTP opening. When analyzing the data observing OSCP in synaptic function, they were able to associate reduced OSCP expression with Alzheimer mitochondrial dysfunction and the synaptic failure that results. They drew this conclusion from the data presented in the rightmost images, which show that neurons treated with OSCP short hairpin RNA show a decrease in synaptic density. And OSCP loss caused a reduction in MEPSC amplitude in OSCP knockdown neurons. So this paper also shows how OSCP interacts directly with amyloid beta. They discovered OSCP amyloid beta bindings in Alzheimer's disease and MCI brains, but not in non-Alzheimer's diseased brains. This also discovered a specificity of OSCP amyloid beta binding, where they had dose-dependent binding, shown by the linear relationship depicted in the left image. This was a direct physical contact. They examined the binding of amyloid beta with OSCP and its affection on function, and found that interactions between the amyloid beta and the OSCP disrupted the ability of OSCP to bind to the F1 subunit. This wrestled and activated MPTP formation. As shown in figure 2, they found that amyloid beta caused more F1 dissociation from the complex, and the F1 monomer identification was verified by the use of an F1 antibody. Altogether, they found that this F1F0 instability due to amyloid beta is partly due to a decrease in OSCP's ability to hold F1 and F0 subunits together. Furthermore, this paper also found that there was F1F0 complex destabilization in the Alzheimer's diseased mice brain mitochondria, and that there was an age-dependent increase in F1 dissociation. So lastly, this led to a strong relationship which connected the deregulation of OSCP to mitochondrial dysfunction. They aimed to see if amyloid beta mitochondrial dysfunction could be reduced through restoring OSCP in mice. OSCP restoration had guarding effects on loss of membrane potential, decrease in ATP production, and MPTP formation, which indicates that OSCP restoration could be protective against amyloid beta toxicity, and similar results were found in AD humans. They also found that when OSCP was overexpressed in AD mice, amyloid beta synaptic loss was improved, but baseline levels of synaptic density were unchanged. When this data was put together, it was evident that amyloid beta toxicity effects on neural, mitochondrial, and synaptic function could be protected through OSCP restoration in mice. So for our conclusions, we found that a major result from this paper includes the finding of OSCP loss and OSCP amyloid beta interaction constitutes the major OSCP alterations in the brain from AD patients and the mice that were used. Furthermore, it was found that synaptic mitochondria are more vulnerable to the amyloid beta-induced mitochondrial alterations, revealing the causative role of synaptic mitochondrial dysfunction in the development of synaptic dysfunction in Alzheimer's disease. This finding allows us to further our understanding of the intracellular influence of amyloid beta toxicity in Alzheimer's. Another critical finding in the study was how reduced OSCP levels and the interaction of amyloid beta with OSCP, which impairs mitochondrial function in AD-related pathological settings, implicates that amyloid beta toxicity induces OSCP aberrations, leading to F1F0 ATP synthase dysfunction in AD-sensitive brain areas. Additionally, it was found that downregulation of OSCP and neuronal mitochondria significantly sensitized MPTP formation, a well-documented key mitochondrial dysfunction in Alzheimer's disease. OSCP restoration reduces amyloid beta-induced MPTP overactivation and preserves synaptic function without having a detectable influence of baseline MPTP formation and function required under normal physiological conditions. So the further research for this paper states that we can explore if any amyloid beta may affect mitochondrial proteins other than OSCP that contribute to mitochondrial F1F0 ATP synthase dysfunction and MPTP activation in Alzheimer's disease. Additionally, evidence has suggested that Alzheimer's disease is a systemic disease as amyloid beta is detected in circulating blood, platelets, and other tissues. In view of the complexity of mitochondrial abnormalities in Alzheimer's diseases, the findings of OSCP aberrations in the study may only provide one mechanism for systemic mitochondrial dysfunction. Because of this, it is important for future studies to evaluate the impact of OSCP alterations in AD on a systemic level. And here are our references. And thank you all so much for listening. Do you all have any questions for us? Thank you.